Okay, I think we'll get started. So it's my great honor to introduce Dr. Sami Farhi today. Sami is directing the Functional Genomics and Multiomics platform at the Broad Institute of MIT and Harvard. So today he's gonna talk about how CRISPR perturb sequencing screening and spatial omics can be adapted for functional genomics. Dr. Farhi received his Bachelor of Science degree in chemical biology from University of California, Berkeley. Following that, he did his Ph.D. training in chemical biology at Harvard University. Dr. Farhi joined the Broad Institute in July 2018 after his PhD study, and then he's developing methods for all optical neurophysiology. He leads the platform for spatial technology, and the goal is to bring the next generation sequencing platform with proteomics approaches to understand large-scale biological problems. In parallel, he's also directing his own research group, which is focusing on the technology development and IPS-based screenings to understand psychiatric disease with optical methods. I had the privilege of meeting Dr. Farhi 4 years ago when I'm in the job market, and I get a lot of wonderful advices and practical suggestions for which spatial technology to use and what questions to address. So I see him as a role leader in the field, and he's gonna share many of the stories today with the audience, and I hope to open up the discussion after the talk. So we will have an in-depth discussion after. But with that, let's give him a round of applause to welcome Dr. Sami Farhi. Thank you.
Okay, am I on? Sounds good. Thank you. Thank you so much for the really kind invitation, and thank you for the wonderful visit so far. It's been shockingly smooth. Everybody's been incredibly kind. It's been a real blast. So today I'm going to tell you a little bit about two stories, maybe 2.5, depending on how the time goes. I will try my best to leave some space because there's inevitably some things that we work on that I'm not going to talk about. Hopefully we'll have some time to discuss if anybody's interested. You just heard a little bit about like a bunch of weird roles and responsibilities that I work on. So I want to give you a little bit of an introduction to my strange little job. The Broad has basically a large organization, which is called the Broad Platforms. There's a lot of people doing a lot of different things, but very broadly, the work splits into innovation work and scalable services. Scalable services probably make sense to you if you think about it through the lens of a core facility. Basically very large scale services, products. You think whole genome sequencing, which we can deploy for both research projects and for clinical applications. And in parallel, we have the mid-stage methodology R&D. So really the goal isn't to be doing I always make the joke with a light towards optogenetics. We're not really trying to go into the Dead Sea and sequence a bacterium and find the molecule for the very first time. We're more trying to think, okay, now that the molecule exists, what needs to do before it's ready for those n equal to 1,000, n equal to 10,000 scale studies? What kind of applications do we need to develop? What kind of hardening does the methodology need to do? What kind of demonstrations do you have to show? And so I have a lot of very talented colleagues who run research teams in many different areas, from genomic methods to genomic perturbations, to CRISPR engineering, to imaging, to data sciences, and of course our group focuses on spatial. And then the idea is that all of this gets funneled into our businesses, so to speak, which operate in 4 or 5 different areas: genomics, clinical translation, and data services. I oversee the bottom row here. I have a group that focuses on spatial omics R&D, and I look over the functional genomics and multi-omics business units and make sure that we're delivering useful things to the world. If you want to talk about any of that or how I've come to do this sort of strange situation, I'm happy to. Just find me, email me afterwards. Today I'll just focus on the R&D because it's a research flavored talk. At the Innovation Center, surprise, surprise, we sit at the interface of two fields. On the spatial omics side, we do things like benchmarking new spatial technologies. We develop new computational methods. We try to take the edge off of some of the challenges that we see in interacting with spatial data, which tends to be very large. And of course, we do a fair amount of hardening of the protocols as they come out of the companies and they need QC metrics, or they need beta testing, or we need applications in challenging tissue types. Those are all research projects that we would take on in this side. And then in parallel, I have a very long-standing interest in functional genomics. I did my Ph.D. in optical electrophysiology, and I've been obsessed with this question of how cells regulate their behavior and how they make decisions to decide their cell states. And so we perform iPSC screens, and we do a variety of phenotypic and transcriptional-based screens, including with my original technology in optical electrophysiology. Naturally, this is suggestively laid out as a Venn diagram because what I want to talk to you today about is the two methodological stories where we integrate both the spatial readouts and the functional genomics aspect by bringing CRISPR screening to spatial omics. The two studies I'll talk to you about are one carried out by my first postdoc, Loic Benan, who is now a faculty member at McGill University, where we use MERFISH, a spatial transcriptomics method, combined with guide RNA detection. And a flavor of the things you'll see is a few warnings. If you're doing scaled Perturb-seq, there's some artifacts we found along the way that may impact your thinking. To show you how this can be used for multimodal phenotyping and how we can use it to recover cell-to-cell interactions in tissue. And then I'll also hopefully have time to discuss a project led by a very talented graduate student, Brian Kang, who is graduating this summer before starting his job at the Arc Institute. He's co-advised with Ernest Frankel at MIT, and he's been focusing on a protein-based approach rather than an RNA-based approach, where we're trying to think about ways to prioritize the phenotypes to screen to make experiments a little bit more tractable. And cheekily, I refer to this as doing pooled screens the wrong way around, and I'll show you what I mean sort of at the half point through the talk. But I want to get right into it. So in the Perturb-FISH story, there was an early realization around 2019 or so that there were, broadly speaking, two ways to do single-cell resolved CRISPR screens, and that these two methods are complementary. The first is Perturb-seq or crop-seq or CRISPR-seq, or it has various different names, but this is probably familiar to many people in the audience. Essentially, you perform CRISPR perturbations inside usually a Cas9 stably expressing cell line. You perform a single cell readout. I would normally say 10x, but now there are other technologies that also allow you to do this. And then you somehow capture the guide RNA identity alongside the gene expression, and you generate this beautiful PowerPoint map of the gene expression versus guide RNA identity. And if you look across the axes, that tells you gene modules, which is to say genes which if you perturb them, they have common mechanisms of action, and gene programs, which are the response mechanism, so the x-axis and the y-axis of this matrix. This has been successful, and right now people are scaling it up to as much as they possibly can to build virtual cell models. But in parallel, there was also another method, which is called optical pooled screening, where it's essentially starting from the same material, which is to say CRISPR-modified cells. But now, rather than using a single-cell gene expression, you're performing in situ optical phenotyping. So that can be something as simple as looking at the expression of, or in the localization of a GFP-labeled antigen, you know what I'm saying, a GFP-labeled transgene, an antibody stain, or it can be more sophisticated, something like a cell painting assay, which tries to do an unbiased morphological readout and generate a fingerprint for a cell. Or you can push this out as arbitrarily difficult as you want, as we have many experts in the audience who know the powers of imaging tools. But the point is that after you perform this phenotyping, you can then do guide RNA readout in situ. So you amplify out the guide RNA. I'll show you some chemistries that make that possible. And then you perform literally in situ sequencing. You crack open Illumina kits. You do the chemistry by hand instead of using the sequencer, and you get several rounds to assign a particular guide RNA to now an imaging phenotype. And that allows you to relate what the cell perturbation is doing to an actual functional phenotype. I've listed a few, I just gave a few examples. You can imagine that this space is very large. But if you want to integrate all three of those, there was no technology that enables all that. That's where Loic's insight came in. He realized that if these are the three things that we want to do at the same time, so that we can link the gene programs and the gene modules, which then give rise to the particular functional phenotype, well, gene programs and functional phenotypes is just spatial transcriptomics. You just need to maintain spatial information, and then you can perform any kind of optical readout that you want before you carry out the spatial gene expression. And the challenge is actually to read out the guide RNA identity. The problem with that is that guide RNAs are very short, so you can't detect them with the standard approaches that we would use in spatial transcriptomics. There's not that many copies of them. And so there was a biochemical question to answer about how we amplify that signal enough so that we can detect it. And I'll give you the insight all in one slide. The insight is to steal, of course. There was a wonderful paper from Mike Elowitz's lab at Caltech, which is called Zombie. In that paper, they showed that if you incorporate a T7 promoter inside the genome, you can then perform post-fixation in situ amplification with T7 polymerase to generate many copies of an mRNA wherever that promoter landed. Loic's insight then was to realize that the space between the U6 promoter driving the expression of the guide RNA and the top box of the guide RNA is just large enough to sneak a T7 promoter. So conveniently, that worked with basically no optimization. So he cloned in this hybrid vector, which is just the U6 promoter T7 guide RNA. We packaged it into lentivirus, the same as you would for any other single cell functional genomic screen. We transduce that into a Cas9 positive cell line. It doesn't have to be Cas9. It works with dCas9. I'll show you an example. Then we let the Cas9 do its thing, knock out the gene, express many copies of the guide RNA. And then after we profile the cell, we fix it. We perform T7 amplification to get many copies of the mRNA. And then we can perform standard, quote unquote, spatial transcriptomics, where we can now detect both the guide RNA sequences with a normal probe and the transcriptomic readout at the same time. So this is a single modality. It's the same molecule, so you don't have to worry about hybridizing two different protocols together and maintaining the signal throughout. The raw data looks something like this. At the top, I'm showing you 15 different images of the 15 different bases we're using to encode a guide RNA identity. There is a big spot, which corresponds to the T7 amplicon. And you see that in this particular case, this guide RNA identity is encoded by a bit that lights up in the 3rd, 4th, 7th, and 10th, 12th image. And then right afterwards, we just carry out, this is the same experiment, just continuing on. We're now probing against the transcriptome, and you see many, many more dots, which are all combinatorially encoding different transcript identities. And so that allows us to link that guide RNA perturbation to the gene expression. So that's it. That's the story. The rest of it is just proving that it works. I'll show you 3 different examples where we try to challenge ourselves. First, we have a proof of concept in THP1 macrophages where we want to make sure that this is performing comparably to Perturb-seq. We carry out iPSC-derived astrocyte experiment where we try to push the technology quite a bit to a more difficult phenotype, a more difficult cell type, more difficult perturbation. And finally, I'll show you an application in xenografts where we show that we can perform we can study interactions in more complicated cellular milieus. So starting from the THP1s, first question is, okay, is this giving similar data to Perturb-seq? So we conducted parallel screens of Perturb-seq and Perturb-FISH. Obviously, we wanted the Perturb-seq data for other reasons as well. But we basically have Cas9 expressing THP1 macrophages. These are an immortalized cell line. We transduce them with two different libraries. On the left-hand side, with guide RNAs against about 3,000 genes. On the right-hand side, with just 70 guides against 35 genes and 4 controls. We really wanted to make sure that we had enough power for this experiment. So we did a very small number of guides, but a large number of cells. The guides are targeting genes in the LPS response pathway. And so then we stimulate with PMA and LPS to activate the macrophages. And then we perform either single-cell RNA sequencing or MERFISH in order to read out the Perturb-seq and the Perturb-FISH data. And so, first of all, we carried out multiple replicates of this experiment, and we found out that the significant differentially expressed phenotypes are highly reproducible. This is 2 separate replicates I'm showing you from Perturb-FISH. You see we have an R of 0.92. That's pretty good in this field. So then we bin these results together, or we combine them together, and we compare them to the Perturb-seq, and we see, yes, indeed, we do have similar log-fold change effects recovered by Perturb-FISH and by Perturb-seq. The correlation is obviously a little bit lower across different modalities, but it's still quite high, especially relative to standard in the field. I'm also taking this opportunity to show you a heat map that will come up a few more times during this story. The point here is that you're seeing the perturbed gene along the y-axis, the recorded gene along the x-axis with a panel from MERFISH. In every single cell, there is an upper triangle and a lower triangle. The upper triangle shows you the fold change with Perturb-seq. The lower triangle shows you the fold change with Perturb-FISH. The point here is that they are generally always the same, at least the same color, if not the same amount. So that's saying that the two methods are highly correlated. But remember this in a slide when that stops being the case. And finally, we just performed a power analysis to see that the number of cells that you need to recover significant effects is very similar between Perturb-seq and Perturb-FISH. So that's all fine, but obviously you don't need to develop a new method to recover something you already know. You want to see if you can find something new. And Loic had an insight that if you just look at any cell culture, essentially, there are spontaneous variations in local density. Some cells have more neighbors, some cells have less. This happens no matter how good you are at cell culture. It's just a stochastic phenotype. And he saw that if he analyzed the gene expression data, just the gene expression data, some genes actually had a density-dependent gene expression effect. So in this case, TNF expression is going up as the number of neighbors goes up. IL-1A expression is going down as the number of neighbors goes up. And so he started to wonder if Perturb-FISH could recover local cellular circuits with just spontaneous cellular density fluctuations. The first way he looked at this is to look at the effects of local density. Basically, are there different fold changes in the effect of a gene depending on whether it's a cell which has many neighbors or a cell which has fewer neighbors? I told you things would start looking differently. So now we're seeing the same graph as before, but instead of showing you Perturb-seq and Perturb-FISH, I'm showing you high density and low density fold changes. And this is my word of warning. First of all, the good news, most of the time it doesn't matter. So regardless of the cell density, we're getting very similar effect sizes and directions. But all of the black boxes that I've highlighted here are the places where the effect size is not just different, but opposite. So that means that if knocking down this gene causes an upregulation at low density, it will cause a downregulation at high density, or vice versa. And there's, it happens 1% of the time, 2% of the time. You're going to see some differences in effects. And this is something I think we need to consider because we don't frequently talk about the cell density that we use when we carry out our Perturb-seq screens. We tend to try to be efficient with our media, which makes sense, but this leaves you exposed to getting bias one way or the other. There's also a more challenging experiment that you can do, which is to see how perturbing one cell affects the unperturbed cell next door. We can do that because we have a lot of non-targeting guide RNAs in our library. And there we again see phenotypes. What I'm showing you here now is on the x-axis, on the y-axis, I'm showing you the perturbed gene, and on the x-axis, I'm showing the recorded genes. These are just the significant effects. but now the gene is being recorded in the cell next door. And you see that there are a fair number of cases where upregulating or downregulating a gene in one cell affects the neighboring cell's expression. And so, in addition to the technological advancements, we're showing you that Perturb-FISH is working CRISPR knockout, and you can recover effects similar to Perturb-seq. I'm also showing you we can recover cell density-dependent effects. And just to give you a flavor for the kind of biology you can find out, for example, NF-kappa B knockout upregulates TNF at low density, but not at high density. Or MyD88 knockout decreases TNF in the central cell, but increases it in surrounding cells. Yes, please.
Like you mentioned, in that experiment, what was the percent infection? Like you mentioned comparing a non-target control to a target.
Yeah, so everything is nominally infected because there is a selection step that I neglected to. Yeah, so yeah, is our detection ability 100%? Nope. So we are looking for specifically the ones that we can tell are non-targeting and the ones that are specifically targeting. And then there's a lot of, mystery. Of course. Okay. So I'm going to tell you about how we're pushing the technology. This example is going to be biologically lame, but technologically interesting. And I'm going to try to save the biological lameness in the back half of the talk. But basically, we have an ongoing project in autism spectrum disorder. We're trying to do functional genomics. We're trying to figure out what are all of the ASD genes that have been recently found, and like recently, I mean within the last 5 or 10 years. Do they have common mechanisms of action or not? And Loic had been doing, just sort of as a community service exercise, he had been helping a postdoc in the adjacent lab carry out some calcium imaging screens in a new model of iPS-derived astrocytes. And while that was happening, a paper came out which suggested that iPS-derived astrocytes can show large activation following ATP stimulation that's in their calcium levels. And that is specifically related to autism spectrum disorder genetic backgrounds. And so we have a cell type which matches what you see from primary astrocytes in terms of the calcium function, which is the very short summary of the many panels that I'm showing you here. And the question was whether astrocyte calcium function could be a point of convergence for ASD risk genes. So the hypothesis was that we were going to knock out a bunch of ASD risk genes. We were going to monitor the calcium phenotypes. We were going to see common responses, which ideally match those that we observe in the previously published study. And then we'd be able to find out which genes are associated with those phenotypes. So this is what we did. We took 125 ASD risk genes. These are from the aficionados. These are from the Satterstorm et al. Paper, married with the Spark prioritized 50 gene list, which comes out to about 125. We performed ATP stimulation, and simultaneous calcium imaging with Fura2. And so you see traces like that. And then we performed Perturb-FISH. We recovered the expected calcium trace clusters, as you see here. And I'm putting a little asterisk on the large peak cluster over here, which is the one that was seen most phenotypically similar to the previously published ASD study. And then here come the heat maps again. I'm showing you perturbation versus gene expression on the big map. But now I can also show you the calcium cluster identity for a particular cell versus gene perturbation and versus z-scored gene expression. And so we can now start exploring the genetic basis for calcium activity. We can see that USP9X knockdown leads to a bias towards the large peak cluster, and that also is associated with differential expression of IL-6ST and LTBP1, which are both calcium binders. So that seems to make sense. Unfortunately, the gene modules don't seem to map onto the calcium enrichment. This is where I said it was going to be biologically unsatisfying. But we still recovered 4 different ASD risk gene modules in the transcriptional data. So that means that, yes, there are common mechanisms of action, It just doesn't mean that the point of convergence is on the calcium phenotype. It suggests that these genes are doing something different. As a technical demonstration, we were still quite happy because we're showing that this works with CRISPR-I, we can work with more difficult cell types and genes, and we can do simultaneous functional imaging. But our conclusion so far was calcium dynamics was not a point of convergence, but there is something to look for, and I'll show you at the back half what we're looking for. At that point, the reviewers decided to challenge us, and we did our best to respond. The argument was like, okay, everything you've done so far is just in cell culture. Does this actually work if there's multiple cell types, if there's something more complicated going on? And so, fortunately, it's important to have good colleagues, and it's equally important to have really good friends. Rob Manguso is a friend of mine from grad school who runs the lab next door, and he studies. He does in vivo screens of cancer. I'm going to do my best to try to explain the system, but basically, he takes an immunocompromised mouse. He injects it with PBMCs isolated from people. He then generates essentially cell line libraries modified with Cas9 and guide RNAs, injects those into the flank of the mouse, and they grow up into essentially a tumor after 2 weeks. By having the PBMCs injected into the mouse, that means that we can now have both human T cells and human genetic background tumor cells in the same system. And so, okay, so we did an experiment exactly like that using the THP1 library from before, again, targeting the LPS genes, but now in A375 melanoma cells. We found first, intriguingly, that we saw much larger effect sizes in A375 cells in vivo versus in the THP1 macrophages. It's not unheard of that an in vivo system would give much larger effect sizes, but they're almost a factor of 5 larger, so that's something to take note of. The more important thing is that now this is a tumor, which is where we can map tumor subtypes and T cells spatially, and we can map the identity of the guide RNAs throughout the whole system. So that finally allows us to do a very similar experiment to what I showed in the THP1, where we're perturbing genes in the tumor cells, and we're monitoring the expression in the surrounding T cells. And so this may be a little bit hairy to look at, but basically the way to think about it is that several of the T cell genes are related to T cell activation. And so we're saying that there are genes that you can perturb in the tumor, which will up and down regulate T cell activation in the surrounding neighborhoods. We have a lot of work to do ahead of us. We have to do multiple replicates. We should probably do this across different models. We should do it with intentionally chosen genes. This is something that we're striving for right now, but at least it demonstrates the kind of things that you can do with this method. And I'll just give you a flavor for the kinds of genes. IRAK1, LBP, and TAB2 knockout in tumors decrease T cell activation markers in the surrounding cells, but CHUK and MAP2K2 knockout increases T cell activation markers. So if you're thinking about biasing things towards immunotherapy, in principle, my suggestion would be CHUK and MAP2K2, with the knowledge, of course, that there's a lot more work to be done before we say anything. And so that's where the story ends for perturb-FISH guide RNAs. I can take questions now, or I can move on to the next step right away. I'll pause so people can decide. Yes, please, Niels.
Do you know why the density of cells makes a difference for the macrophages?
No, I mean, I don't know. I can guess. So the question was, do I know why the local density makes a difference in the macrophage results? And the honest answer is no. I suspect that there are secreted factors which can be picked up locally, and that if I had a sensitive enough assay, maybe a single molecule assay, maybe I would be able to detect those things. But I don't actually know.
Do you know what the distance is over which those factors would be traveling, diffusing? I mean,
we don't have enough power to properly analyze that. I can tell you that the definition of neighbor was basically either 5 or 10 microns away, so 10 microns away centroid to centroid. So I seriously doubt we would have enough power. Like we could probably do an RNA scope experiment on the single condition and try to get at that, but we haven't done that experiment.
Hey, so in the cell density effects again,
Yeah.
did you detect gene modules that possibly link to something like signaling?
No, we didn't have, like, there wasn't obvious clustering. There weren't that many, like, there weren't enough effects to do any kind of clustering. It was like, you know, like I said, 1 or 2% of the effects at all were giving anything. So, no, I don't think, I don't know that we could have detected that. I think we need a lot more power. The right way to do that might actually be to just, now that Perturb-seq is super cheap, maybe the move is to just do high density, low density Perturb-seq and just subtract the two results and see what happens.
Got it. And in your xenograft study, did you identify any neighborhood effects of the tumor with the immune cells?
Yes, so that's what these are. So these are all neighborhood effects. So the tumor is affecting the T cell in that neighborhood. Yeah. Yes. Yeah. No. So like the problem is your power collapses like I don't know if it's quadratically or exponentially, but like fast as a function of distance. So it's already kind of a stretch. Like you see how many. I'm filtering out all the non-significant effects here. And so you can see how much smaller this heat map is relative to the ones I was showing before. That's not an accident. That's because we're probably leaving a lot of signal out. And I don't know if the negative results are truly negative or we just don't have enough examples to see them. These might just be the biggest effects. Yes?
The technology looks beautiful. Did you include any of the perturbed genes among the monitored transcripts? And if so, what's the variability in CRISPR efficiency? I mean, you're subsampling already, but are you subsampling for the cells where you are confirming CRISPR?
Yes. So we, not in the THP1 library, so not in example 1 and example 3. In example 2, yes, we are seeing, we have the genes. Our efficiency of detecting the knockdown is not as good as we would like. This is actually, you know, it happens pretty frequently. So the things that made it onto the heat map or didn't make it onto the heat map had to have correlated effects against the guide RNAs from multiple targets, rather than confirming that it was a knockdown of the target. This is, you know, look around. I think you'll see that a lot of the Perturb-seq studies have a similar challenge, that they can't actually detect the downregulation of their own target. So that could be poor guide RNA design, but I don't think poor guide RNA design is going to have the same off-target effects on the same gene. So, yes. I just don't think we have the cells. And we also don't have the genes. Remember, you do those kinds of analyses when you have all the genes and you're looking for enrichment. But in this case, we have a very narrow set of genes, so it's about 500 or so. So if you don't need to enrich, it's either there or it isn't. Something that we're going to try out hopefully in the near future is using more whole transcriptome level effects, and in that case, yes, that would be a very fun thing to do. But you know, those technologies are just coming out now, and so we'll have to wait a few months.
Do you see a dosage effect in gene editing? Does the cell take 3 or 4 edits, or does different cell types take different preferential guide RNA?
I'm not sure how to. So we don't notice it because we haven't looked for it. I don't know the way that I would have done that. You know, nominally everything has a single copy. Like everything is a single copy transduction throughout the whole study. So in principle, there should only be one guide RNA. How many actual edits is that one guide RNA doing in every cell is something that a lot of people would like to know the answer to. There was just 2 papers published. One of them is a preprint from Ramnik Xavier, and there's another preprint that another Nature paper that I embarrassingly don't remember the author or the name. So I'm sorry, I'll just have to leave you with that thing. But where they're directly sequencing the edits in those particular cells, and they can see those responses. But the problem is that in those cases, like it's like $4 a cell. And so you really got to want to know the answer to that question. And it's not really compatible with screens of this scale yet.
Let's keep moving.
Okay. All right. 20 minutes. We will do the short version of this talk. Now remember, these two stories were happening in parallel. Brian was thinking about neurodegenerative disease, and he was asking himself, like, how do we map large numbers of genetic hits in neurological disease to specific pathways? And like the traditional approach, in air quotes, is that you would take a list of risk genes, you would perform a phenotypic screen, you would hope that something comes out, and then you would cluster these into pathways somehow, maybe on your previous knowledge, maybe on some biochemical work that you do afterwards. But the problem is that the phenotype choice is hard. You saw an example of that earlier on, where we chose the wrong phenotype. There are many established neuronal phenotypes and astrocytic phenotypes, but it's unclear what their disease relevance is. And one way that you can get around finding pathways is to perform very large-scale studies and then perform guilt by association. So that says, well, this risk gene has a very similar phenotype to a gene that I know what it does, or a gene whose pathway I'm familiar with. But the problem with that is that you need a lot of scale because you want to go ideally genome-wide. And the neurons are pretty sparse, particularly when you go to IPS systems. And so it becomes very difficult to do financially, logistically, experimentally, everything. And so Brian had a different approach, a different idea, which is to start from all of the genes, just in the transcriptome, nominate pathways enriched for risk genes based off of prior knowledge. This is all computational. Then, based off of that, prejudge what phenotypes might be interesting, and then come around and do a very small focus screen to validate all of that and confirm that the pathway is accurate. So we called this selfie because it reminded us of turning the selfie camera around. It was a backronym also. And I'll just walk you through a very quick example of this whole study. I'm going to fly past the computational side because that is why I have very talented collaborators in Nurse Frankel. I will be taking no questions on this side. But basically speaking, we're focusing on Huntington's disease. There are many different data sources that one could use, including human genetic studies from the Kindred cohort, a GWAS study, which all modified the age of onset of the disease. And at the same time, there are in vivo and in vitro models for neurodegeneration in wild-type Drosophila, and in Huntington's disease specifically, there are knockout screens in vivo in mice. And so Brian used a tool that was developed by Nurse Lab many years ago, which was called Omics Integrator, which takes all of these hits and projects them onto existing protein-protein interaction databases. And uses something called the price-collecting Steiner-Forest algorithm to maximize to basically pick out which pathways are most likely to be relevant to the particular disease. This basically minimizes the noise in the interaction data and maximizes the chance that you get pathways with disease relevance. And so that pulls out, when applied to Huntington's disease, that pulled out a very large set of genes which are interacting, and I should say proteins really, proteins which are interacting with a bunch of like previously annotated functions based off of geo-ontologies. And I'll show you, overall there were 931 putative genes which like modified the Huntington's disease age of onset with 23 different subnetworks in neurons. And I'll just show you, we're going to focus on one specific one, which is neuron projection development. Which looks like this, and you're seeing the interaction between genes which are known to give earlier and later age of onset from the GWAS studies, as well as some hits from the Drosophila study, which seem to be interacting and seem to be on neurodegenerative pathways, as well as some genes which don't come up from any study that we put in, but are predicted based off of the interactions from the protein-protein interaction. So we call those predicted nodes or Steiner nodes. And you can see IAR5 and GAS7 in here. So now that we have that, the question is like, okay, is it real? Are these real pathways? Do they actually interact? Do they have an effect on neuron projection development? And to answer that, Brian turned to a protein-based approach. We stole, again, a method called Perturb-map from Brian Brown. I should say he actually shared the constructs with us, which encodes guide RNA identity using a series of small epitope tags. So if you think guide RNA1 is encoded by having a flag tag, an HA tag, and a CMYC tag, for example. Guide RNA2 has flag tag, HA tag, and an S tag, for example. And that way we can differentiate different guide RNAs. This allowed us to then perform multiplexed immunofluorescence. We used Codex or PhenoCycler, depending on how you know it. We're using procode antibodies to both identify the guide RNAs, and because these are trafficked to the outside the cell, we're also using them to map the neurite projection maps, as well as a set of phenotyping antibodies which label different cell organelles, which we're using to perform cell painting type phenotyping to generate a morphological fingerprint. And the idea is that the neuronal arborization antibodies illustrate a use case where we know what the phenotype is, and we've naturally selected cells where we know what the phenotype, or genes where we think we know what the phenotype is. And the morphological fingerprints are for when we don't know what the fingerprint is or what the phenotype is, and we just want to go unbiased towards the study. This is what the data looks like. So these are the procode maps. You can see the different dots show you cells lighting up in different rounds, very similar to how we were encoding guide RNAs in the RNA space before. And these are the phenotyping antibodies that we used, including a set of stress markers and a large set of organelle markers, which are chosen to be very similar in staining to cell painting dyes, which are commonly used in the field. Brian then used a maximum likelihood algorithm to predict which guide RNA, which cell is most likely to be associated with which particular procode identity, and by extension, which guide RNA. And long story short, the measurement of the guide RNAs looks something like this in 7 colors, and we can now predict the actual known gene knockdown in more specifically like this. We performed, we segmented all the soma out, we extracted features from the multiplexed IF of the cell painting, and we could perform differential feature expression, confirming, for example, that FAN1 knockdown has a large number of Golgi-related morphology changes. This is just encouraging us that things are working. And then, of course, this tracks with what a visual inspection of the images. This is the point I really want to make. There were now a set of clusters that emerged. This is a pairwise AUC. Basically, the closer you are to 0.5, the more likely it is that 2 genes have the same phenotypic effect. And we saw that there was an enrichment for things that were close to each other to genes that were predicted to be next to each other in the protein-protein interaction network. So essentially, I'm saying that we're confirming our predicted network based off of genes that are having close to interactions, having similar morphological phenotypes. We also could use the same method to trace the neurites, as I discussed earlier. So I'm going to fly through this just to get to the final picture here, which is that now we're looking at the neurite length, number of branches, number of trunks. These are conventional neuronal phenotyping features. And we can see, for example, that our HRAS and ARPC, which have a decrease in neurite length and an increase in number of trunks, show also quite similar features and quite and in both the soma features based off of the multiplexed IF and in the neurite features extracted from more traditional methods. A similar story can be told from the other cluster, from SLC25A11 and SCCAP1. Again, we see that those genes are very similar to each other in the soma space and in the neurite feature space. And as an added bonus, SLC25A11, also the effects we're seeing also match what we expect from the human genetics, and that both of these genes should have the same direction effect, even though they're coming from two different genetic sources in the original input. And so I wanted to just get to this conclusion slide. Basically, in selfie, we're letting computation in PPI and then guide the choice of phenotype first. And then if the pathway is real, it should give similar phenotypes in a very small screen. And then very relieving for me, morphological fingerprinting of the soma give very similar results to the targeted phenotypes of the neurites. This is important because if you're a neuroscientist and I tell you about cell morphology, you're usually thinking that I'm talking about neurite arborization. But what our results suggest is that actually there is enough information in the soma to similarly predict those arborization phenotypes. And that in turn gives us a license to scale up the work, because it's a lot easier to measure soma features in an optical pooled screen than it is to measure neurite features. 2 o'clock, so I have 10 minutes. Okay, so regretfully, I'm just going to give you a highlight. I would love to discuss this more with you. We've now taken, based off of this, we've taken the cell painting features and we've performed an optical pooled screen just with the soma localized features for a much larger set of genes, 443 autism spectrum disorder genes. This is the set of genes that Loic tried to find a phenotype for before, and we didn't find one. Now we've performed fingerprinting, and we found 7 different clusters among 150 odd genes. Several of these clusters make sense. They include targets of mTOR and TSC1, which we recovered just based off of morphological similarity to each other. And when we dive into it, we see that the targets of mTOR and TSC1 are indeed up and down regulated within our phenotypical panel. And then when we dived in a little bit further and we carried out these measurements at both day 4 and day 28 and day 10, we see that there's an advancement or retardement of the maturation of the neurons in these ASD clusters, suggesting that there are sets of ASD genes which converge on different morphological phenotypes, and that these clusters track with the established neuronal maturation reports. And indeed, we're finding larger numbers of genes which haven't been reported to affect neuronal maturation. And most excitingly, about 2 weeks ago, my dear friend Xin Jin published a paper doing in vivo Perturb-seq, and she's finding many of the same genes showing up as clusters in a mouse model of ASD, suggesting that we are onto something in terms of finding real sets of genes that work together. But I will stop here so that we can discuss. You can ask me questions and you can tell me that none of this makes sense. Basically, my overall argument is that you can get a lot out of even small scale screens. They're better for tests of hypotheses. They allow for exploration of more conditions. I want to remind people that spatial methods are particularly powerful in internally controlled experiments, which is important while the methods are so expensive. And finally, that a lot of the things that make this possible is that we're relying as much as possible on single methods and internally and commercially available systems. So I'll stop there so we have time to talk. I just have to acknowledge my whole lab, and specifically Brian, Loic, other Brian, and Ernest, who are the lead authors and the co-PIs on these studies. So I'll stop there and I'll take questions. Good.
Do we have questions?
Yes.
I can do both.
Please, I'll repeat it for the people on Zoom.
For the second part,
Yeah.
Any of the hits seem to have components of the
SNF complex. Did I look at this right?
No, you saw it exactly right. Yeah. Yeah, it is.
Wondering if in your screen that you laid out,
yeah,
there be the next iteration of it. If we could just look at the nucleus and features of it in there, would that give us also similar effects in terms of what would be happening for your phenotypes in the soma and in the neurite?
So we are probably already doing that. It's just that we're not as smart as you are. So the question was, we're getting a lot of SNF hits, and if we restrict our analysis to just the nuclear features, does that still pop out? So the answer is that our fingerprint includes the nuclear features explicitly. We're just combining all of that together. We're actually doing I should just look. There's a figure right there. There is, we can separate out the features based off of which channel they come from. And so the nu-n channel would be a great one to look at because that's mostly nuclear. I don't remember if that cluster is enriched for nu-n features. I can find out. Just send me an email. I'll let you know. Yes. Hi, Niels.
Really nice talk. When you say, and maybe I didn't quite get this, the nearest neighbors showed up in the perturb analysis because they have similar phenotypes. But that was in a case when you had known the pathways. So how much can this actually be reproduced if you don't know pathways to find new pathways?
So that's kind of what we're doing. I mean, I guess I would argue that that is what we're doing here, because these are now ASD genes where we're not prioritizing them based off of having similar phenotypes. We're essentially just using that entire first half of the second part to justify looking at this phenotype as a neurologically relevant thing. But here, these are just ASD genes. I mean, one hopes that they're on the same phenotype, but we don't know, or on the same pathway. But the clustering suggests that they are on the same thing. And in fact, if you look, we are recovering genes that are known to act together. So mTOR and TSC1 are going in opposite directions, and TSC1 is an inhibitor of mTOR. Yeah.
I guess also my question was one step further. Can you also build the connectivity back and say, not only they belong together at some point, but this must affect this closest neighbor, that only then affects the next.
So the question is, can we build the connectivity map just based off the morphological features? That I feel less confident in. I would probably do that with Perturb-seq rather than with, Brian thinks yes. I am not so convinced. I think that will probably be very difficult, and it will probably be easier to just run the Perturb-seq screen because you can be on the same pathway a little bit or a lot, and it's the magnitude on that pathway. It doesn't really tell you where you land up or down stream of that. And then you're going in opposite directions depending on if you're an activator or inhibitor. So all of those things I think complicate that.
Sorry, quick question.
Please.
So are these features cell profiler features or vision transfer? Okay, thank you.
Yeah, it actually kind of, Brian loves all different kinds of ML tools. So we've tried DynoVT and it's 6 other ML methods. It's just what he likes. I can't stop him. But they all do the same thing. It actually kind of doesn't matter what you do. Pretty much. It's also very difficult to say, what does better mean? Because the definition of the ground truth is cell profiler. There's no such thing as ground truth for morphological fingerprint. And so sure, the best you can do is say that they're similar or different from cell profiler. So it's chicken and egg a little bit. Yeah.
I have a technical question for the slide containing codecs.
Yeah.
When you're choosing codecs, what is the design rationale for choosing codecs over SCSAP or other like Cosmax?
Cosmics didn't exist when we started the study, so that was a strong motivator. The only options were PhenoCycler, which we had because Aviv had already bought one before I had joined the lab, and that was a pretty strong motivator. And it was less work than setting up a SCSAP panel. I'm sorry, but that answer is so unsatisfying. If I was doing it today, I would use Lunaphore. And we actually have shown that we can use it works just fine, and it took us less time to develop the panel. Yeah.
To conclude, I have one last question, General. As a platform director, what is your view for the volume of different technologies, and what would be the general advice you give for people who are mumbling about choosing a technology?
Well, my coward's answer is always depends on what the question is. I would say for the median user, our recommendation right now is, whatever time it is today, my recommendation for most people is to use 10x Xenium. There's a few assumptions in that. One is that you don't have infinite money, and two is that you don't have infinite time. The reason for that is that the 10x has done a really good job of making sure that their method is about equally performant on almost everything we throw at it. And so if your friend got some results that you liked from 10x, you have a reasonably good chance of also getting good results from it. I can't say the same thing for other tools in the space right now. I've made a large investment in Merscope, larger than I would like to think about right now. That has not paid off to the same degree. But my mouse brain work on Merscope is totally fine. So your mileage may vary. If you need sequence specificity, if you need direct sequencing, well, then I'd start asking you questions about what kind of samples you have. If you want to know SNPs and FFP tissues, I will probably start breaking bad news to you, because I think that's very unlikely to happen with the kind of resolution and sensitivity that people generally want from these analyses. I think that data is just not, I think the biomolecule is no longer in the cell by the time that question is asked. Most other things, there's probably a way. Again, it depends how degraded the sample is and what the specific question is.
Fantastic. I don't see any Zoom questions. So let's give Dr. Farhi another round of applause. And thank you for being here.
Thank you.
For the wonderful talk. Thank you.